Hey everyone, welcome back to BI23. In the last video, we ran AlphaFold 3 on the official server. In this video, we are going to run AlphaFold 2 using something called ColabFold, and we will do it completely free on Google Colab. No installation, no complicated setup. Just open, paste, and run. Let us get into it. So what is ColabFold? ColabFold is a simplified, faster version of AlphaFold 2 that runs inside Google Colab. Google Colab is basically a free online notebook where you can run code without installing anything on your computer. Someone has already written all the code for us. We just need to fill in our protein sequence and click Run. That is it. Nothing else. Here is what we will do today. First, we will open the ColabFold notebook on Google Colab. Second, we will enter our protein sequence. We will again use Human Insulin so you can compare the results with AlphaFold 3. Third, we will run all the cells and wait for the prediction. And fourth, we will download and look at our results. Let us jump into the tutorial now. Okay, so first open your browser and search for ColabFold AlphaFold 2. You will find a link to the official ColabFold notebook on Google Colab. It is also linked in the description below. Click on it, and it will open in Google Colab. Now, before we do anything, go to the top menu and click Runtime, and then click Change Runtime Type. Make sure it is set to T4 GPU and click Save. This gives us a free GPU to run the prediction faster. Now, you will see the notebook has several sections. Each section has a play button on the left side. We do not need to write any code at all. We just need to fill in one box and then run everything. Scroll down to the first input section. You will see a box called Query Sequence. Click inside it and paste your Human Insulin FASTA sequence that we downloaded from Uniprot in the last video. If you do not have it, just go to uniprot.org, search Human Insulin, click P011308, and copy the sequence from there. Just below that, you will see a job name box. Type Human Insulin here. No spaces, just one word. Keep all other settings as default. Do not change anything else. Now go to the top menu and click Runtime, and then Run All. This will run all the cells one by one automatically. You will see them running one after the other with a loading spinner on each one. Now here is something important. The first few cells will install the required tools. This takes around 3 to 5 minutes the first time. Do not worry if you see a lot of text and code running. That is completely normal. Just let it go. After installation is done, ColabFold will start the actual prediction. This is the step that takes the most time. Around 5 to 10 minutes depending on the server. You will see a progress bar and messages like running MSA and running prediction. Just wait for it to finish. Once it is done, you will see a message saying the job is complete. Now scroll down to the bottom of the notebook and you will see some output images appearing directly in the notebook. The first image you will see is the coverage plot. This shows how many similar proteins were found in the database for your sequence. More coverage means AlphaFold had more data to work with, which usually means a better prediction. For human insulin, you will see good coverage since it is a very well-studied protein. The second image is the PLDDT plot. This is the same confidence score we saw in AlphaFold 3. High values above 70 across most positions means a confident prediction. For insulin, most of it will be high confidence just like before. The third thing you will see is the 3D structure shown directly in the notebook. You can rotate it and look at it from different angles. Just like AlphaFold 3, the colors represent confidence. Blue is high confidence and red or orange is low confidence. Now to download your results, look at the left side panel in Google Colab and click the folder icon. You will see a folder with your job name, Human Insulin. Right-click on it and click Download. It will download as a zip file just like AlphaFold 3. Unzip it and you will find your PDB structure file and all your result images inside. Keep this folder safe and you can access it anytime. Now quickly, what is the difference between what we just did and AlphaFold 3 from the last video? AlphaFold 2 via ColabFold is fully open source and free with no usage restrictions. It is great for predicting single protein structures and is the most widely used version in research today. AlphaFold 3, on the other hand, can handle proteins together with DNA, RNA, and drug molecules, which makes it more powerful for drug discovery. For most everyday bioinformatics work, AlphaFold 2 is perfectly fine and is what most researchers use. And that wraps up our complete AlphaFold series here on BI23. We covered the theory in Part 1, ran AlphaFold 3 in Part 2, and now AlphaFold 2 in Part 3. Coming up next, we have Nextflow, Snakemake, Phylogenetics, and a lot more. So make sure you are subscribed so you do not miss any of it. If this video helped you, please like and drop your questions in the comments. Happy learning! See you in the next one.